IL-6, and others. This is all studies that have been done on the basis of these variables. That basically, if you see these variables, that means there is inflammation in the system. And what we have found is, for example, Nakagawa offered the first direct observation of unstable free radicals that were generated by laser performed by EPK at 100K. And in 2012, research by a number of investigators such as Kuller, Tomuk, Obsur, Sahin, Sepisi, Alton, and Seyman, and a lot of others, has concluded that radiofrequency induces biochemical damages by increasing free radicals, and the free radicals attacks to structural biomolecules such as DNA and lipids. Researchers reporting increased inflammation, as you can see, are all of this and a lot more. Chushi Nogawa, Nasiski Kiro, a lot of these names I have a hard time pronouncing. I have to apologize about that. But you can take a shot of this slide and you can search them, or I can provide you with a research if you email me. Researchers, we have found an increase of inflammation under the radiofrequency. Martina Bruschmann, Christian Wolpert, Thomas Birch, and a lot of other researchers, all MDs. Now, there is a necessity for non-traumatic technologies. Why? Because we want to decrease inflammation. If we're looking at an anti-aging situation, if we're talking about anti-aging, and if inflammation increases aging, we need technologies that decrease inflammation, not technologies that increase inflammation. Now, we can increase fibroblast secretion of collagen without any trauma. We can do that by utilizing specific signals delivered in specific frequency combinations. And this is about the most famous protein that all the lasers are talking again and again, radio frequency, that they're increasing collagen. But we can do that with certain signals. You don't have to traumatize the body to do that. So Lossinger et al., since 1999, and Yang et al. In 2004, have shown that there is a significantly higher production of collagen at very specific frequencies, as well as changes in the dynamics of calcium, suggesting the involvement of ion channels. Now, signaling technology is a new technology that is developing. I mean, if you subscribe to Science magazine, you're going to see many articles, hundreds of articles on signaling pathways, how the different cells communicate with each other. I will try to introduce you into this technology a little bit. It's quite complex and not easily understood, but I will try my best. But it started primarily with neurophysiology research in natural frequencies produced by certain parts of the brain. And then in cardiology, of course, the pacemaker. That's signaling technology. What is the pacemaker? It gives the signals of pacemaker cells that are not there anymore. In brain research, external frequencies were built to stimulate and reinstate natural frequencies of brain in lesioned areas by the process of resonance. And this will bring us to kind of newer, more complex technologies that we're going to talk about eventually. Utilizing signaling pathways to reduce inflammation has been now a new approach. Now, this involves research on signaling pathways and experimental studies and results on rejuvenation and on decreasing inflammation by these signaling, manipulating signaling pathways. Now, what cells do, they basically integrate different signals. Like our brain, when it hears different sounds, integrates these sounds into sequences that are understood by us as speech, as language. This is what the cells do with the signals that they receive. So the proteins in each cell receive a complex combination of signals which simultaneously trigger many different signaling pathways, which provide an opportunity for crosstalk between different signals. Now, signaling includes different cell-to-cell communications, and this slide shows how basically communications happen. And all these light up areas are basically signals for communications that are happening at that time. Now, there are different functions that the cells fulfill by these signals. So one is extracellular matrix communication, and this is, you can see here how this can happen. All these lines that combine these two cells are sort of a communication network that is formed, just like the internet very much, or other communication media. Now, also, the cells are leading to different cellular responses, which one is adhesion, another one is migration essential for tissue formation in wound sealing. So different cells migrate in another area to kind of heal the wounds. Proliferation, this is how cells increase, basically, and differentiation. Now, you cannot have proliferation without differentiation. It won't work. Proliferation without differentiation is going to give you a tumor. It's going to give you a cancer. So that's what cancers are. They just proliferate, but they don't differentiate, not like the normal cells do. They do have a cellular differentiation, but not quite. Now, what differentiation means is that after the cells proliferate, they become different cells. They become a muscle, for example, they become blood, they become a nerve cell. So this is the cells prior to differentiation, and this is the cells after differentiation. So, but the problem is that the greater the complexity, the larger the possibility of communication mistakes. And the way we understand today aging and illness is that it's nothing more, nothing less, but a communication mistake. So clusters of multiple ligands and signaling receptors interact with each other, and specific cellular responses result from the integration of multiple incoming signals. And any aberrant communication between cells and their surroundings, extracellular matrices, can lead to a plethora of human diseases. And for example, atherosclerosis, arthritis, renal lac fibrosis, and of course cancer. Aberrant communication between cells and the surroundings and extracellular matrices also leads to aging. Now, signaling, how is signaling decoded? How is there a way of decoding signaling? Yes, there is. The way we do it is signaling decoding is relied on simultaneously crystallizing and measuring multiple proteins in thousands of individual cells. So you do that at the same time. And this is how you look at signals. That's how you decode the signals of the cells. Now, perturbing these cells with molecular interventions drives the ordering of connections between pathway components. So different cells you take, is the way we speak, just always keep in mind the frequencies of spoken language and how these are combined into words. So it's just sounds. You combine this into words, and these words have a meaning eventually. It's the same thing. It's exactly the same thing. It's like a complex computational process. And to decode that is another very complex computational method that automatically looks and kind of decodes the signaling relationships. Now, reconstruction of network models from physiologically relevant primary single cells can be now applied to amplify faded configurations of signaling pathways to ultimately replace dysfunctional signaling in aged or diseased cells. How is signaling decoded? There's more. Another body of researchers is exploring the resonance that is mediated. Basically, as the cells communicate, there's a sort of a resonance. If the two of them click, there is a resonance that happens. That means resonance is amplification. That's what resonance is. So resonance-mediated increase of signals is involved in the synthesis of certain proteins, such as collagens, elastin, hemoglobin, actin, myosin, adipase, that's the energy that forms the ATP, hormone secretions such as DHEA, that is for the skin and the bone, T3, that's for lipolysis, and IGF-1, that is the hormone that builds the muscle, responsible for the muscle. Now, what is the purpose of decoding cellular signaling? What we want is we're trying to form a universal language of our cellular microcosmos, like a new language, the language of life. And here is how you can see like an insulin code. This is the code of the insulin. As you can see, it starts with a sequence of ADG, and it ends in AGG, and then there is another sequence and which ends in ACC. This is like letters, and basically this is a code. It's how you create, this is how you would write it, for example. But in fact, it's a conglomeration of different frequencies. Now, missing signals can leave the cell with incoherent instructions, and therefore they're going to give inappropriate responses, and that will be deleterious to the cell. This is what another theory of cancer is, that it's the wrong signaling. Now, investigating how such signaling pathways are organized is relevant not only for understanding how normal cells work, but also for appreciating the molecular basis for aging and disease, since aging, as well as many human disorders, result from breakdowns in signal transduction. Now, this is a research that was done on cells, and it was done on the basic research by Xia et al. That aging is the result of protein communications that have diminished. So here, out of 4,000 different proteins that were identified, these proteins comprised about 7,103 protein-to-protein interactions, almost twice as many. And the way it's done, it's not in a sort of a linear fashion, but it's sort of in a very complex way, where one protein gives signals to many other different proteins. So what you see is that result of examining all of these interactions, the 7,103 protein-to-protein interactions in aged versus young subjects, what this demonstrated is that aging is the result of the disorganization in this interaction. So the more disorganized these interactions are, the more there is aging. Now, in Cambridge, the research looked at signaling properties of young versus aged cells, and then tried to sort of transfer the signals from the young cells into the aged cells, and what they found is a sort of a rejuvenation, if you like, of the aged cells. Now, ion channels are crucial in signaling. Ion channels are also intricately related to a number of biological functions. For example, calcium signaling has been proved to be strongly associated with aging, manic depressive illness, and neurodegeneration, or optimal neurological functioning. And in stem cells, what we have found, very interesting part, is that when the potassium channels are blocked, and this is Johns Hopkins University research, what you see is that you see no differentiation in stem cells. And if stem cells do not differentiate, like I said before, they will form tumors and then cancers. So potassium channels are crucial in stem cell differentiation and absolutely crucial in stem cell therapies. Now, another very interesting fact that came out from